mean, I started this by saying, Derek, that I was going to get it so that you understood exactly what we're talking about here. So I think
let's do it.
We're a little too far
down there.
down the road, and what I think is going to happen is that you may end up still being confused. And I don't want that. I want you to know exactly what we know so that you can decide for yourself who's correct. If we could do that.
I'm following you so far. I want to just really be able to outline this, and I do want you to go down that road, Tom, because I know that I have listeners who are already on board with you guys, and I have listeners who think that you're crazy, and I have listeners who have no idea what's what. So just that's why I'm asking these very simple questions to get to distinguish between purification, isolation, and I definitely would like you to. You said you can make it as simple as possible. So please, I just wanted to clarify that. Definitely we have audiences coming at this from different.
Right. So if Derek, I could use you as a foil here, and somehow Andy is frozen. So I don't know if that's a problem, but so let's just take this step by step, because that's my method of understanding the world. So the first thing is, and again, I would appreciate if I say anything and you don't agree or don't understand, that you stop me and so we can clarify. Is that okay?
Sounds great.
Okay. First of all, can we agree that a virus is a thing? And what I mean by that, it's not a thought or a feeling or an esoteric idea. It's an actual physical thing.
That's what I've been taught, yes.
Okay. Now, let's take another example, the one sort of Andy said. And I want you to pay attention to the words that I use, because it's important. So we want to do an experiment to see whether the caffeine isolated from a coffee bean causes high blood pressure. So in other words, we want to isolate in the usual sense of, I have a pencil here and I isolate the pencil, so that's the only thing I have is this pencil, right? You with me?
Yep.
So we take the coffee beans and we grind them up and we put them in a capsule and we give it to 10 people, and then we give an appropriate control, and all 10 people have high blood pressure who took the ground coffee bean. Have we now proven that the caffeine in the coffee beans causes high blood pressure?
I guess if you're saying you're controlling for other factors, then you probably narrowed it down at least.
So I would question that, because I don't think you know that the only thing in ground up coffee is caffeine. In fact, I would actually submit that until you prove that, there's likely to be fibers and caffeic acid and aromatic oils and a whole host of other things.
That makes sense. Yeah.
So we need to take this a step further. So we take that ground coffee bean that has a lot, maybe even hundreds or thousands of different things in there. And again, our quest is, does the caffeine in coffee beans cause high blood pressure? So we put it through a filter paper, and that collects all the fiber and the cells or whatever else is in the ground part. And you end up with what we call coffee, right? And then we give that to 10 people. We give them an appropriate control. Have we now proven that the caffeine in the coffee causes high blood pressure?
Sounds like you're getting closer.
We're getting closer, but I think, again, correct me if I'm wrong, but I don't think there's anybody who thinks that the only thing in coffee is pure caffeine, right?
No, I wouldn't think so.
Right. And certainly I'd love to see that proven. So again, we have to take it to the next step. And so now we can put it through a centrifuge or flow cytometry or some sort of device, which we can easily do, and we can spin it out. And so you get a band that contains only caffeine. Now, the next thing you do is you send that to an independent lab and you tell them the steps that you did. I ground the coffee at 10 RPMs a minute. I put it through a filter paper and the pore size was this. And then I spun it again in a density centrifuge and it created this band, and I sucked the band out with a pipette. I looked at it under a microscope and I looked at it in a chemical analysis, and the only thing I have in there is caffeine. I can prove it. And 10 other people who all do the same thing will find the same single molecule. And then I give it to 10 people. They all get high blood pressure. 10 who don't, give them a placebo. It's a controlled experiment. They don't get it. Have I now proven that the caffeine in coffee beans causes high blood pressure?
I would think so. I mean, that sounds much more rigorous than where we started.
Yes, I would agree. Now, what about, Derek, if I took the coffee and I mixed it in a vat that has chocolate and tea and yerba mate, and there may be other things that have caffeine in it, and I swirled it up and I cultured it for a day, and then I pulled out the caffeine from that, would I still be able to say that the caffeine from coffee beans causes high blood pressure?
No, I wouldn't think so.
Because you don't know where the caffeine came from.
Exactly.
So that's what I mean. That's how a human being understands isolation. And when I say that's how a human being understands isolation, I would go so far as that is the only way to isolate. Now, it's different. Like if I want to isolate a pen from a pencil, I don't have to do all those steps. I can just remove the pencil and I have an isolated pen. And then I can study the pen. Now, I've looked at 31 different studies, although it may not be the right number because some of them I may have done twice or I might have missed some, all who said they've isolated this virus. And then I heard the other day, 125,000 times it's been isolated. I can tell you, and I am willing to, at this point, retract my book if any one of them did that procedure I just said, which means you take the snot from somebody with COVID and you grind it up just like you do with coffee. And then you put it through a filter paper of known pore size. And that's called the supernatant. And then you put that in a density ultracentrifuge device. And it comes out in a band. And then you suck out the band with a micropipette and you show that that's all you have is this one virus. Then you inoculate or you expose an animal or a person to that one virus, which you now know you have nothing else. And that experiment has never been done. Now, it was done in the 30s and 40s when they invented the the electron microscope. And what they found is this isolated, purified particle didn't cause any disease in any animal or person they studied, not once. And if anybody can show me a single study showing that that definition, which is the common human scientific definition of isolation with a virus causes disease, I will retract the book. Now, how did these people, these papers that you showed, do that? So here's what they did. They take somebody with COVID sometimes they just take a culture. They grind it up, just like the coffee bean. They put it in a filter, and then they have this unpurified brew, and then they stick it on a monkey kidney tissue, and nothing happens. So then they starve the tissue, and nothing happens. And then they poison the tissue with gentamicin and amphotericin, and then the tissue breaks down into thousands or hundreds or millions of particles, all of which have the same genetic material as the so-called virus, because there is only one, building block of genetic material. It's like putting something in a vat of caffeine. And then they say, this particle is the new virus, and it's absolutely pure nonsense. And I hope after that, Derek, you can then read a paper. And if you see that they took the supernatant from ground up cells or snot, that's not purified. And if they inoculate it onto kidney cells, tissue, and it breaks down, and then they take some pictures of it showing a bunch of stuff in the background, That is not what any normal human being calls isolation and purification. And every single one has been that way. And therefore, the challenge stands. Show me, why can't they just take somebody, grind up their snot, filter it, centrifuge it, show us the picture, put it on an animal and show they get sick? Then I can go home and garden for the rest of my life instead of doing this nonsense.
I appreciate that breakdown, Tom, and I know you had something to add, Andy. So I just want to kind of say about that. So is it simply a matter of semantic word games and, you know, spells, as you kind of put it earlier, or is it literally that in the field of virology they define it in a different way?
No, it's not that at all. It's that they discovered, after 20 years of proper isolation, like I described, right? They took chickenpox lesions, they ground them, they filtered it, they centrifuged it, they found it. It doesn't even mean actually that it's a virus, really. It could be something from the inside. But let's forget, that just complicates the matter. And then they inoculated it on animals and they didn't get sick. And they essentially at that point said, we should go home, there's nothing to this, let's be plumbers. And then they did a analysis and found that plumbers make a lot less money than virologists. So they decided to do what Andy described, which is to put it on noxious tissue cultures. And this paper that Andy appropriately describes, when you read Ember's paper, he says, actually, we have no idea whether these particle breakdown products are even coming from the viro, meaning the kidney tissue, or from the outside. They have no idea of that. And so this is the whole shooting match. This is everything, because if this is based on a fraudulent understanding of science and biology, then you can't, and Andy could describe exactly how they go from this to making a, quote, theoretical genome of a, quote, theoretical virus. It's scientific nonsense. I don't know how else to describe it. And then I'll let Andy go. Excuse me for going on so much.